We will start with the nucleus. I think we did one or two topics of nucleus, but I would like to start it again because it will be in continuation if I start it again with you. We had done just about the what is nucleus, who discovered, then we also did the definition of it and what type of cells, if the nucleus are present in the cells, the position, the shape of the nucleus, etc. we did. But I'm going to repeat it for you so that you understand it. Now, nucleus was discovered by, it was discovered by Robert Brown. Robert Brown was the person who discovered the nucleus. Now, when we speak of number means how many it is present in every cell, or whether it is one or two or more than that, or sometimes the nucleus is absent also. So, first is we'll discuss about the cells where the nucleus is absent. So, nucleus is absent in RBCs, and the RBCs which are mature. In immature RBCs, these are present, but in mature RBCs, the nucleus is absent. Then, if we talk of one nucleus or uninucleate, if a cell is uninucleate cell, uninucleate cell means when in a cell one nucleus is present, and such type of most of the cells are like this. Most cells are uninucleate. Then when we talk about binucleate cell means where two nucleus are present. So when we talk of two nucleus are present in paramecium. If you remember paramecium, how did it look like? It was like this. And we had two nucleus here. One was a big size kidney-shaped nucleus and one was small. So this is micronucleus. The small one is micronucleus and the large one is meganucleus or macronucleus. Anything you can say, mega or macronucleus. So binucleate is paramecium. When we talk of multinucleate, cell, when we talk of multinucleate cell, the striated muscles have multinucleate nucleus. They are multinucleate striated muscles. They have many nucleus. Now, there is two terms which we use related to multinucleate nucleus. We either say cenocytic, COENOCYTIC, cenocytic, or we say syncytial. Cenocytic or syncytial. When we say cenocytic, this we say in case of plants. If the plant cells, they have many nucleus, we say that it is cenocytic. If the animal cell has many nucleus, then we say that the animal cell is syncytial. So there are two terms we use differently for the same thing. I mean, two terms are used for the same characteristic property. When a plant cell is multinucleate, we say cenocytic. When an animal cell is multinucleate, we say that it is syncytial. Now let's talk about the position of nucleus. The position of nucleus. So I'll erase this for you. Just excuse me for a minute. Yes. Let's talk about the position. When we say position of nucleus, position means where does it lie. Position, the first type of position of nucleus is centric. Centric. So first type, on the basis of position of nucleus, first type of cell is centric cell. Centric cell means if this is the cell, the location of the nucleus is here. So we consider this as centric. That means centrally located nucleus when the nucleus is present in the center. And this type is present in animal cell. The example of this type is animal cell. The second, on the basis of position of nucleus, is eccentric. ECENTRIC, eccentric. This is, suppose this is a plant cell. So what happens? We have big vacuoles. So the nucleus, it pushes to one side, and this is seen in plant cell. This is the second type on the basis of presence of nucleus. Then we have peripheral. The third type on the basis of position is peripheral. This happens in the case of adipocytes. Adipocytes. We have done adipose tissue. Adipose tissue is the fat tissue. So these are also known as fat cells. They have all globules in them. So if this is a cell, it will have a big oil globule in it. All globule present. This is a big oil globule. And because of the presence of big fat globule or oil globule, the nucleus is shifted to one side. So this is nucleus. And this is fat globule or oil globule. And this is a adipocyte. So because of this, it is shifted to one side. Then we have basal cells, basal position of nucleus, basal, B-A-S-A-L. Basal means at the base, and where it is present in columnar cells, columnar epithelium, columnar epithelial cells. So how do they look like? When we see, it is the columnar is somewhat like this, column-shaped cells. And they have their nucleus located at the base. So this is basal position on the basis of the presence of nucleus in a cell. So we have four types. We have centric, eccentric, peripheral, and basal. Now, any doubt in this? Any doubt in this? Yes, beta. Anything which is not clear to you or any doubt in this? Okay. So this is clear to you all? Fine, beta. Fine. Okay. Now, bache, I'll move on to the next. Just a minute. Yes. So I'll move on to the next, and the next topic is the shape of nucleus. On the basis of shape of nucleus, the first is circular. Most of the cells, they have circular type of nucleus when the nucleus has this shape. So most of the cells, they have circular shape of the nucleus. Then we have another shape which is oval, O-V-A-L, oval type, oval shape of the nucleus. This oval shape of the cell is present in smooth muscle. After oval, the third type of nucleus is bilobed. Bi means two lobes are present. So the shape of the nucleus will be, it will be just two lobes, one lobe and another lobe, like this. Only two lobes are seen, and this is seen in eosinophils. Eosinophils. Eosinophils are a type of WBCs. This is a type of WBCs where the bilobe nucleus is present. After bilobe, we have trilobe nucleus. Cortex, trilobe, where three lobes are present. So same way we will have the three lobes, the nucleus of three lobes here like this. This is present in basophils. Basophils also, these are a type of WBCs. After this, we have multi-lobe. Multi-lobe means more than three. That means it is somewhat like this. This is seen in neutrophils. Neutrophils are also a type of WBC, a type of WBC. Then we have one more shape, which is horseshoe shaped of nucleus. This is seen in Vorticella. So this is what about the shape of the nucleus. These many shapes of nucleus can be seen in various types of cells. So we have these shapes of nucleus. Anything in this you want to discuss, you want to ask? Yes, beta? Anything in this you want to discuss or ask? Yes, bache? Okay, I think this is clear. We come to the structure of nucleus now. The next topic is structure of nucleus. Structure of nucleus. How do the nucleus look like? If we see the nucleus is like this, so this is the structure of a nucleus. Nucleus is double walled structure. So we have the structure of nucleus. Now in this, if we see that, just a minute, what is attached to the nuclear membrane? So we have some structures like this, which are attached to the nuclear membrane. We can make few over here also. Then we have one more structure which is attached to it, which originates from it, is this structure. I'll make it just I'll make it only this much to show you that what is this. So like this, we have the endoplasmic reticulum which is attached to it. Besides this, we have a dark colored body in this, and this dark colored body is nucleolus. Then we have the chromosomal material and we have the nucleoplasm. I think this picture is clear to you. Yes, beta. This is looking clear to you, beta. Is it looking clear? Picture of nucleus is clear. Okay. Now, if you see here, this is the outer membrane. This is the inner membrane. This is endoplasmic reticulum. This is ribosome. These are ribosomes. This brown substance in this is nucleoplasm. These ones, these are chromatin fibers. I'll make it here. This is nucleolus. Okay. Now, few things which I want to tell you about this is, these are these structures, these openings which are present here. These openings are nuclear pores. The special features of these nucleopores are, these are octagonal in shape, octagonal in shape, these openings. And besides being octagonal in shape, these nucleopores have a diameter, the opening of the diameter. That means if we see how big pore it is, so diameter of the opening is, of the pore is about 800 angstroms. 800 angstroms is its diameter. And if we see in the nucleus, how many nuclear pores are present? If we ask this, so in number, their number is about 1000 to 10,000 in number. They are 1000 to 10,000 in number. That means in one nucleus, I made here only 3, but it has many sides. So every side, they are about from 1000 to 10,000 in number. The diameter is 800 angstroms, and these are octagonal in shape. Now, one thing which I want to discuss about these nucleoplasm, the nucleoplasm, I'll make an arrow and write here, it has enzymes. Enzymes are present in this. And enzymes for what? Enzymes for the process of transcription. Transcription you will not understand right now, and translation, because these are the processes of protein synthesis which are there in class 12. So it has enzymes for transcription as well as translation. In besides this, this is also rich in divalent cations, divalent ions like Mg2 plus, Mn2 plus. So this is rich in this also. So this is the structure of nucleus. I think this is clear to you. Ribosomes are present attached to the nuclear membrane. The endoplasmic reticulum is also seen attached to the membrane. Besides this, we have few more structures. Nucleolus, which is a darkly very condensely packed and a darkly colored, darkly stained organelle, which is seen inside the nucleus. Then we have chromatin fiber, which are uncoiled, long and thin. But when the when the cell division starts, then they become short, stout, and condensed. Then we have the nucleoplast, which has many enzymes and some divalent cations also. The nuclear pores are about 1000 to 10,000 in number in a nucleus. Their diameter is 800 angstrom and they are octagonal in shape. Anything which you want to ask me in the structure of nucleus, or shall I continue with the next topic? Yes, beta. I think this is clear to you. Structure of nucleus, bache. Okay, Priya Yadav is saying yes, it is clear to her. Any query in this? You can ask your questions also. If you are unable to understand anything in this, you can ask your questions also. Okay, Siksha, it's clear to you. Rohan, okay. Fine. So this is clear. Now after this, I would like to start with the next topic. And that next topic is, okay, children, I would like to discuss one thing with you. In the test, there was a question on mechanism of insulin action. But in that you have written it in the form of paragraph, although that day, the day a day before exam, when I took your doubt session, in that I showed you that diagram on NCRT book. It was about the mechanism of action, how the hormones they enter, whether it is a protein hormone or a steroid hormone, how they enter a cell. That diagram and explanation of that diagram has to be given, and you have written something else. I told you about insulin that day in the doubt clearing session, that there are receptors which are present on the membrane and how the insulin molecules are received by them. That was told by me, but none of you have made that diagram and none of you have written about the action mechanism of action of insulin, although that was made clear in the doubt session. I showed you the diagram also in the book, but nobody has answered it that way. Yes, beta. Why was that not clear to you? Yes, bache. Yes, bache. Was that not clear to you? What was the problem that all of you have done that question wrong? You all are unmute, beta. I have unmuted you all. You can speak up. You can unmute and speak yourself. What was the reason that you were not able to answer that question properly? None of you have answered that question properly, and I was really, because that question was, your doubt was cleared a day before. I couldn't do that question because I was left with just 2-3 minutes and I had to upload many answers. So I didn't attempt it. Okay, some of you have left it and some of you have not done it. Okay. But because I'm telling you, because that question was told to you in the doubt session also. But none of you have done it. That's an important question. Mechanism of hormone action is an important question. How they differ, if it is a protein hormone or if it is a steroid. That you have to go through that again. This is a very important question. Okay. And if you don't understand, if you are not able to understand after this chapter, I'll take some of those chapters also, whichever you want me to, any other chapter, even from the book, any chapter which you want me to take up, I'll take it with you, but your doubt should be clear. Okay, so now I move on to the next one, and this is about something about the nuclear pore. So for that, I'll just erase this. I have to make you understand. Just little of this here. Okay, I'll make you understand here. I have magnified the pore so that you understand it better. I have magnified the pore, and in this pore, where there is a pore, it's not that there is nothing in between. There is a structure which is present in here. This is a structure like this, and this structure is known as annulus. It is like a ring. It has an opening in the center. This opening in the center, it's not that it is complete open. This structure is known as annulus. A N N U L E S, annulus. This structure is known as annulus, which is present. Now we have here where this membrane is, the inner membrane is, one more membrane, a thin membrane is also attached here. It is present near the, like this, like this. I'm making the set only one. One more inner membrane is present, this, near the pores, starts from the pore till the next pore. Okay, and this membrane, which is here also I have drawn this membrane. So this membrane is known as fibrillar lamina. I'll write here the name, fibrillar lamina. This is known as fibrillar lamina. This particular membrane which is present on the inner side of the on the inner side of the inner membrane. This is outer membrane. This is inner membrane. I've written here inner side of the inner membrane. We have this fibrillar lamina. Now, if you see this complete structure, which is annulus, this complete structure which is present on the at the pore, this structure which covers the pore here, this annulus, the width of this annular complete width from here to here, or I'll make it on the lower side, this complete width of the annulus pore here, from here to here, it is 800 angstrom. That is why we say that the width of the nuclear pore is 800 angstrom. Now the center, the lumen of this annulus, I told you that it has a tube-like structure, this tube-like structure. Actually, it's not that the complete annulus is open. It is open only from here to here. This particular structure is open. I'll make this annulus with another color so that you are able to see it. I'll make it with another color. Wait. I'll make this annulus with another color. Just a minute. I'll make it with red color. This annulus, which has an opening here and an opening here, this is like a tube which is in the center of the annulus. I think this is visible to you. Only this is the place through which the substance they come out, and this is filled with a substance. A gel-like substance is present inside this structure, and this gel-like substance which is present here is known as nucleoplasmin. I'll write it. This gel-like substance, I'll write here, nucleo. Okay, you will not be able to understand if I write it inside. So I'll write this gel-like substance here. I'll erase this. This gel-like substance which is present inside this is known as nucleoplasmin. And this gel-like substance which is present, this through this gel-like substance only this gel-like substance, nucleoplasmin, which is present inside the annulus, this helps in the movement, the transport of substance from outside to inside and from inside to outside. So all the molecules, all the substance which have to be transported either from inside the nucleus to outside or from outside the nucleus to inside, it is through this gel-like substance which is present in the annulus and is known as nucleoplasmin. Anything which you could not understand in this? Yes. See, children, the diameter of annulus is 800 angstrom. That is why we say that the nuclear pore is about 800 angstrom. But one thing is there, the complete annulus is not helping in the transport of substance. It's only this the center tube-like structure which is helping in the transport of the substance, which has this nucleoplasmin in it. Only this is helping, and this particular structure of annulus, the lumen of which is present in the lumen of the annulus, the diameter of this is about 500 angstrom. Actually, the substance is transported only through this 500 angstrom tube-like structure which is present in the lumen of the annulus. But we say that the diameter of nucleus is 800 angstrom because the complete diameter of this annulus, see, the complete diameter of annulus is 800. That is why we say. Anything in this which you could not understand? Yes, beta. This particular fibrillar lamina which is on the inner side of this is rich in electron rich protein. So this has high quantity of electron rich protein. Now anything which you could not understand in this pore, or if you want me to repeat it again, I'll repeat it for you. Yes, beta. Is this nuclear pore structure clear? Okay, fine. Any query in this, or any question or any repetition which is required in this? Yes, everybody should speak. Yes, everybody should understand. There are 13 students today. So, Sepak, Yashika, Sanvi, Lalit, Ananya, Rohan, Priya, Deepika, Zainab, Siksha, Amrita, Harsha, Ananya. Any query in this? The structure of nucleus or the nuclear pore, anything is which you could not understand? Yes, which you could not understand. Clear? I'm getting the message that it is clear. Okay. So children, I think this is the nucleus. This much is okay for today, and tomorrow I'll